Hi, my name is Charlie Ye, CEO of mFluiDx. Our mission is to fight epidemics with rapid molecular diagnostics. Several weeks ago, we sent our co-founder to Brazil to the epicenter of Zika outbreak. What she found was shocking. Within one single lab, there were 3 ice chests like this, full of thousands of backlogged patients waiting to be tested for Zika. Most of these samples will not be tested. Even if they are tested, it will take weeks or months for results to get back, which will not make a difference for the patients or for outbreak containment. Without rapid, accessible diagnostics to determine who is infected, we cannot implement control measures properly. And this is exactly what happened with Zika. Within a year, it spread like wildfire. The scary part is Zika is not the first, nor is it the last infectious disease to hit us. Within the last century, we had hundreds of outbreaks, most of them concentrated within this decade. We had avian flu, influenza, SARS, Ebola, and now Zika. With modern air travel and rapidly evolving pathogens, epidemics continue to spread. Unfortunately, current solutions are not adequate. Dipstick assays, similar to your pregnancy test, are not sensitive enough to look at early detection. On the other end of the spectrum, PCR, highly sensitive, but it's not portable, and it is also very expensive. Costs 10 to 100K. Because you can't bring PCR near a patient, it can take weeks to get the results back. Isothermal amplification is also as sensitive as PCR, but it's also costly. We need a new solution that is low cost and sensitive that could be brought to the field. Our solution is a fast chip. It is so small that you can literally put it in your pocket and bring it out to the field, the fast chip. This chip was originally funded by the Gates Foundation and DARPA with the aim to bring the lab into the field. It is fast. Within 30 minutes, we can test for results. Patients no longer have to wait for days and weeks to get the results back from centralized labs. It is fast. F stands for field ready. The entire system is portable. The user simply opens a vacuum seal pouch, loads the sample onto the chip, puts the chip onto a heat pack for 30 minutes, then puts the chip into a reader, and then uses a smartphone to take an endpoint result. What sets us apart from all the other microfluidic technologies is our core vacuum-based fluidic actuation technology. Without using any external equipment, pumps, power sources, controllers, computers, we can run this chip. This is why this chip is so small and ultra low cost and ready to be brought to the field. It is affordable. We plan to use injection molding to make these chips and readers. We estimate at scaled production, it will be orders of magnitude lower cost than the current PCR solution. Sensitive. We are using isothermal nucleic acid amplification to test for DNA and RNA. Isothermal amplification has comparable sensitivity to PCR, which is the gold standard. We can run quantitative tests on this chip, useful for applications such as HIV viral load monitoring. For example, the more green dots you see, the higher pathogen concentration in your blood. As another mode of using this chip, we plan to run hundreds of simultaneous reactions on this chip and screen for multiple diseases in one single chip. For example, if you have cold symptoms, one chip will tell you which kind of bacterial or viral infection this is, so you can have proper treatment. Trackable. We use a smartphone app to analyze the results, aggregate the results, the GPS coordinates, the patient data into a cloud-based tracking system. This becomes a very powerful disease tracking platform. Ministries of health can use this data to determine where they should put their resources first in the hotspot. Our go-to-market strategy is tiered into 3 stages. Initially, we'll be selling this as a research tool. First, as a PCR alternative, then sample prep module. After we integrate these 2 together, and after we get FDA approval, we'll market this as a point-of-care diagnostic assay for humans. To validate the market, we've talked to OBGYNs, local doctors, hospitals, and researchers, and they all express strong interest for this technology. There's also interest in mosquito surveillance to test mosquitoes for dengue and Zika to track diseases. We have licensed 4 patents from UC Berkeley. These patents cover the fluid actuation, the overall system design, and the patterning on chip. We have a strong founding team. Dankar, our co-founder, was the original lead inventor of the TB Cepheid assay, which became one of the world's leading point-of-care assays. I was the original inventor of the FAST chip, which is funded by Gates and DARPA. We are working with world-class collaborators, the first scientists to discover Zika in Brazil. Here's a video of our field study.
So we need to develop this technology before the next outbreak so that we're prepared to go right into the field and contain the outbreak.
Tiene que ser un diagnóstico rápido, sensível e barato. É importantíssimo para evitar a disseminação da doença.
We've developed a test to diagnose people anywhere, in the field, in a clinic, in their homes. It doesn't need any power, do molecular detection of DNA or RNA of different infectious diseases.
Esse equipamento não precisa de técnicos super treinados. É a eficiência do teste e sua alta qualidade. Estamos todos satisfeitos com os resultados encontrados.
We need to make sure that this project gets off the ground and that it works properly and as expected with high sensitivity and specificity in all situations. And so we are asking for funding so that we can continue this work.
As you can see, there is a huge need and huge market for diagnostics. But why are we personally motivated to do this? Dankar has worked in the field and experienced firsthand the heavy cost of delayed response. As for me, even at home, when I go to the doctor, I like to see my results right then and there and not be anxious about it. Imagine how much more Zika patients must anguish while waiting for their backlog results. We have technology in our hands that can change how diagnostics is done and save millions of lives. We invite you to join us in this mission and fight Zika and the next 100 outbreaks. Thank you.